Yo, what is up, guys? Welcome back to another um, update to Road to Medical School. This time at number 5. So we're gonna go over some stuff that I did. So I kind of been uh, just moving around. So I made a pretty huge decision. I changed my major to public health, and I'm gonna want to focus more on the healthcare system, just study about it, and also having time to study for the MCAT because that's going to be a huge portion. Because I already know I'm not exactly the best student out there, so I just know that I got to basically lock in for this. So we're going to start off with some stuff that I've been reading from the MCAT biology books, especially the Kaplan books. They're really great. They really dumb down the topics for you, so you really understand. It's like this, compared to the Princeton books, the Kaplan books are way better in my opinion. I've tried looking at the Princeton books, and I have to be honest, I like the Kaplan books a lot better. So let's start off with what we're going to talk about: prokaryotic and eukaryotic and viruses. So basically, it all starts off with the cell, right? Cell is the most basic form of life. Is the most basic form of life. Let me get my paint out here. Right, the most basic, very basic form of life. Right, cell. Oops, let me fix that a second. Get this up. I don't have an iPad or anything, so I can't really show you guys. So, cell is considered the most basic form of life. Usually has a nucleus for eukaryotic cells, but there's a nucleolus in the middle. That's where DNA is formed. Right, there's a membrane. You got a membrane as well. So, anyways, so our human body is just to clarify first. So MCAT is designed to test you on a whole variety of knowledge such as biology, organic chemistry, biochemistry, chemistry, behavioral sciences and psychology, physics and statistics, and the infamous CARS section, aka critical analysis and reasoning skills. Right. So the MCAT doesn't. The whole point of MCAT is not. You don't. Yes, you have to memorize a ton of stuff, but it's not something you really focus on. You want to be able to apply these things to actual problems and concept. You need to understand the concept, not just memorize a bunch of things. Because you understand the concept, everything else will be a piece of cake, right? Because when it comes to medicine, you're learning about the human body and diseases and viruses, right? And we're gonna eventually find out that viruses are actually not living things. So, yeah. So there's many cells because you go from cell, tissues to organs, organ system, and then all organ systems make up the human body, right? So now the first thing to consider is cell theory. There's just three basic uh, forms of cell theory, but there's also a fourth one that was later added. So the thing is that all living things are composed of cells. That's true. Me and you, we're both made of cells, and cells are the basic functional unit of life, and that cells arrive from previous existing cells. So this cell here came from a cell, another cell before it, and that cell before it, and on and on and on and on and on, right? Now a fourth one was eventually added, where basically it found out that cells carried DNA inside them, DNA, right? DNA, and this DNA can be passed down to the other cells through mitosis, meiosis, stuff like that, right? So now we're gonna go over eukaryotic cells, the organelles inside them. So one of the big differences between eukaryotic cells and prokaryotic cells is that prokaryotic cells do not have any membrane-bound organelles. That's probably like the main huge difference. Okay, so eukaryotic cells, they their nucleus is enclosed in a membrane, while prokaryotic cells do not even have a nucleus, right? So there's quite a few organelles that we have. The Golgi apparatus, endoplasmic reticulum, ribosome, cytoplasm, mitochondria, lysosome, what else? Centrioles, the nucleus, and of course, it's not really a organelle, but we got the vesicles, and then the cell membrane, which is a selectively permeable phospholipid bilayer, embedded with proteins and sodium potassium pumps. Which yep. So so the membranes are like in a low state of cytosol in the cytoplasm that basically lets them be afloat. So let's say we got a nucleus right here. It's the surface of the cell membrane, right? The surface of cell membrane is no, hydrophilic, but the inside are hydrophobic. So phospholipids are stuff that looks like this, and then you got the little tails, right? Like this. You got little tails, like that. That's the the membrane, the cell membrane, or most membranes look like that, right? Oops. Didn't mean to do that. Where's the back button? Here it is. Alright, cool. So DNA stands for deoxyribonucleic acid. It is where our genetic material is encoded, which are later organized into chromosomes. Well, I'll go over that maybe next episode once we start looking to reproduction. And so all eukaryotic cells are reproduced by mitosis, while meiosis is through sexual reproduction, right? So we'll go over the nucleus first. So nucleus is surrounded by a nuclear membrane or an envelope, and it's a double membrane. So a majority of organelles are double membrane. The only exception is the lysosome. Lysosome is a single membrane. So, and on the nucleus, they have these things called nuclear pores that allows for two-way exchange of materials between the cytoplasm and the nucleus, right? Now, DNA contains a little coding regions called genes, and those DNA is able to be organized into histones, which are later turned into they're wounded up and turned into strands called chromosomes, right? So I'm not gonna go too deep in that. So basically, all I know is that nucleus is kind of like the brain of the cell, but it's also where the genetic information is stored. And then there's also a subsection of the nucleus called the nucleolus. This little tiny balls inside here. That's where our RNA is synthesized or made, called ribosomal RNA. Again, not gonna go too deep into that. So now go to the mitochondria. So the mitochondria is by far one of the most important and complicated organelles in the cell. Without it, we would not be able to do anything, right? So a mitochondria is uh, also a double membrane organelle. So it's often called the powerhouse of the cell. You probably saw a lot of memes about it. How like yeah. So it contains an outer and inner membrane, and the outer membrane is like a barrier between the cytosol and the inner environment of the mitochondria. Well, the inner membrane, this thing right here, is like folded in little flaps or little foldings called cristae, Cristae, and that's where all the stuff with the electron transport chain happens, as well as all the little stuff like cellular respiration. Everything goes on in the mitochondria. So the whole point of the mitochondria is for cellular respiration to create ATP, adenosine triphosphate, right? So, and there's a little space between the inner membrane and outer membrane called the intermembrane space, and it's something just called mitochondrial matrix, right? That's all it is. And what else? So yeah, so mitochondria, they're a lot different from other cells because they're considered to be semi-autonomous, which means that they reproduce through binary fission. So a lot of bacteria are kind of like this as well. So they basically go through asexual reproduction when the cell splits into identical daughter cells, right? And it's also this is theory of how the mitochondria came to be. So it was an idea that a long time ago in aerobic prokaryote, when an ate was eaten, no, it was eaten by an anaerobic prokaryote, which resulted in a symbiotic relationship. So a long time ago, uh, was a cell, but was eaten by another cell, and it basically just lived inside that cell, and eventually evolve into an organelle of its own. So, kind of interesting about that. But mitochondria are also, instead of their not only job is also provide keeping energy, because what I do know is that about 50% of all energy, ATP created from the mitochondria is used just for the sodium potassium pumps inside the cell membrane. All right, what else we got here? Look at show my notes. So, and also your mitochondria was given to you by your mother, right? Because the reason for that is because the sperm from your dad does not have a mitochondria. So, yeah, mitochondria are also able to kill the cell by releasing enzymes from the ETC, electron transport chain. This is called apoptosis or cellular suicide. So now next organelle is called the lysosome. Lysosome. So the lysosome is they look like little cups, I think, like little cups, right? Like this, so for example. I'll call this a lysosome. We'll draw two for the hell of it. Lysosome, like that. We're gonna color code this. We're gonna make this green just cause it's more uh like that, and then we'll color code lysosome to brown. And for nucleus we'll make it purple. Right, so like that. Okay. So anyways, uh give me a sec. Make sure how much time we got left. Okay, good. Okay, so lysosomes are they break down stuff, right? So they contain hydraulic enzymes that basically break down different substrates in the body through endocytosis. Basically, they kick that stuff out of the cell. So, and they sometimes work with endosomes, which basically transport, package, and sort stuff in the cell, and they go through a process known as autolysis, where they similar to mitochondria, where they can uh, destroy cells by using their own enzymes. So it's similar to mitochondria. It results in apoptosis, which is cellular suicide. So just know that lysosomes are kind of like, I guess, the cleanup crew of the cell, I guess. So now we're gonna go through something a little more complicated, the endoplasmic reticulum. So there's two different parts of this. There's the rough and smooth ER. So the rough one kind of looks something like this. Let me see, get a pin out like this. A bunch of foldings like this, right? Foldings, and it's also embedded with proteins like that. Embedded with proteins. It's really bad drawing, but whatever. Anyways, so like that, right? The rough ER is studied, no, not proteins. It's studied with ribosomes. Ribosomes, my bad. Sorry, guys. So, a ribosome is what allows the translation of proteins. So it has these things are ribosomes, which lets you have protein synthesis, right? So smooth ER is a little different, right? Smooth ER is usually like next to them, right? It's next to it. It's on the side, on top of it, like this. This is the smooth ER. That. And it's smooth, so the reason why it's called smooth is because it's got these little bumps and ribosomes on it. So their job is lipid synthase and also for detoxification of certain drugs and poisons. And they also transport proteins from the rough ER to the Golgi apparatus, which we'll talk about next. So, the Golgi apparatus, it looks very similar to the smooth ER, so I'm going to draw that in blue, in this weird color. As soon as the Golgi apparatus. Okay. So, the Golgi apparatus is kind of seen as what I like to call it, like the FedEx of the cell, the UPS, the cell. They have they're made up of a bunch of membrane-bound sacs, and they basically transfer anything from the endoplasmic reticulum, from the both rough and smooth ones, are then transferred to the Golgi apparatus, where they are packaged in things called vesicles. And then once inside the Golgi apparatus, they're modified by adding stuff like carbohydrates, phosphates, sulfates, to to modify it, and then once they modify it, they send them out in vesicles, and then once the product is good to go, they leave the cells to exocytosis. Exocytosis is the cellular process that moves large molecules out of the cell and into the environment. Right. So now we got peroxisomes. I actually can't remember what they look like. So let me look them up. Peroxisomes. So they're also another organelle. Okay, I can't really find a picture of it. Alright, we're gonna draw them as circles for now. We're gonna draw them as little circles for now. Let's draw two for the hell of it. You can make them red. Whoops. Not me to do that. I got red. So, peroxisome contains something called hydrogen peroxide, or H2O2. So one of the main functions of peroxisome is the breakdown of the fatty acid known as fatty acid through B oxidation. So what beta oxidation is a catabolic process in which the fatty acid molecules are broken down in the cytosol in prokaryotes. That happens here in the mitochondria, while in eukaryotes it happens uh, yeah, no. For prokaryotes, it's cytosol, while in eukaryotes, it's mitochondria. And this whole process of beta oxidation is to generate acetyl-CoA, right? So, other than that, the peroxisomes they are in charge of the creation and synthesis of phospholipids, and they contain some enzymes that are in charge of the pentose phosphate pathway, which we're going to talk about later, but that's more of a biochemistry thing. So anyways, now on to the cytoskeleton. The cytoskeleton is relatively complicated. There's a lot of moving parts to it. It's about 3 parts to it. Right, so we got microfilaments, microtubules, and intermediate filaments. I'm going to just draw that on the side, okay? So, make the background like gray or something. Make more shine. Right, so, and then we can try to draw here what this thing here looks like. Circle. So, it's like lined up like this. Like that. Imagine a bunch of circles like that, and then it's like the phospholipid tails like that. Right, it's also embedded with like those proteins like sodium potassium pumps that let stuff move in and out of the cell wall, stuff like that, right? So anyway, so microfilaments are made of actin, right? So the majority of everything else, they're kind of have they use tubulin instead. So microfilaments, they're part of the structural protection of the entire cell, and they often cause muscle contractions through myosin. Myosin is a protein that's for muscle contraction. So, and they also go through they also play a huge role in cytokinesis, which you're gonna later see in mitosis, which is basically the division of material between the daughter cells, right? And also during mitosis, there's something called the cleavage furrow, which is formed from these microfilaments, which basically acts as a ring where the site division between two new daughter cells occurs. So go on to microtubules. Microtubules are like the hollow polymer of tubulin proteins, right? And they create pathways for both kinesin and dynein to carry vesicles. So kinesin is a motor proteins that move organelles and vesicles along the microtubules in the eukaryotic cell. While dynein, I think that's what it's called, it uses chemical energy from ATP to make uh, make make long microtubules and transport cellular stuff, basically. Yeah, and microtubules also help with the structure of cilia and flagella. So here's where it gets kind of complicated with the cilia and flagella. So it, so basically, The microtubules, they have a structure known as the 9-2 uh 9-2 doublets. How do I say this? The flagella has a thing called 9-2 structure where they have 9 rings of doublets with 2 microtubules in the center. I'll try to draw that out somewhere in the corner. How do you make a circle? Like this? Like that. 1, 2, 3, 4, 5, 6. 1, 2. Alright, pretend there is 18 of them here, right? Pretend it's 18. Just assume it's 18. And you got 2 in the middle. Like that, and then they're all connected in lines. Like they're in pairs, so like that. Line there, line there, line there, line there, line there, uh, line there, line there, like that. And then here, they have these little things called the inner sheaths, which looks like this basically. And then these things also have another more things that connect sticking out of it, known as the dynon arms. Right, like that. So yeah, this is known as the 9-2 structure. 9-2 structure. Right. Now, it's a lot different for centrioles. So centrioles, they have 9 triplets of microtubules, right? So they organizing centers for microtubules, and our structure was a hollow center instead of the 9-2 structure. And they also help with organizing the mitotic spindle. So that's for mitosis, like the PMAS stuff. It's like prophase, metaphase, anaphase, telophase. So next up is the intermediate filaments. They're a diverse group of filamentous proteins, which includes keratin, desmin, vimentin, and lamins. So they are involved in cell adhesion and just overall integrity of the cytoskeleton. Right? They're just kind of there, to be honest. So I'm not gonna go too deep on it, but it really depends on the cell structure. Like microfilaments made of actin, while microtubules are made of tubulin. So yeah, not gonna go too deep into it. Now we're gonna talk about tissue. So these cells, they eventually will turn into tissues, right? They eventually will turn into tissues. So tissue formation is a unique characteristic of eukaryotic cells only. So prokaryotic cells, they can't do this because they're considered to be single cellular organisms. So there's really 4 main types of tissues: epithelial tissue, connective tissue, muscle tissue, nervous tissue. Nervous tissue would be something like stuff like your brain. Muscle tissues, like your heart stuff. Connective tissue can be stuff like your bones, while epithelial tissue can be something like let me think here, like the nephrons in your kidney, stuff like that. So, anyways. Or epithelial, it's the main tissue that covers your body in all the cavities, like the pleural cavity, all the junk and bunk. So it's the main protection you have against pathogenic invasions and desiccations. So the epithelial tissue is like the main part of the parenchyma. So the parenchyma is like, that definition is called the functional tissue of the organ, right? So most epithelial tissue are polarized, meaning that one side faces like inwards towards the organ, while the other side of it faces the outside, where it interacts with the blood vessels and all the other cells. And there's quite a bit of organization for this. So we got simple epithelia, so it only has one layer of cells. Then we got stratified epithelia, which has multiple layers of cells, right? Then we got pseudo-stratified epithelia, which is basically It looks like simple epithelia, which means one layer of cells, but it's actually stratified epithelia. So it looks like it's multiple layers. So it looks like only one layer, but it's actually multiple layers. So, and of course, can't forget about the different shapes. So there's cuboidal, there's cube shape, columnar, long and thin, squamous, flat, and like kind of like a fish scale, kind of square like. Anyways, so we're not go too deep into that. Anyways, so connective tissue, they basically support the body and the framework for all the other epithelial cells, and they're the main ones that form the stroma, right? And they support structure by secreting materials to form extracellular matrix, right? Some of that includes stuff like collagen or elastin. So some examples of connective tissue would be like your bone, cartilage, ligaments, fat tissue, or like adipose, and your blood. Your blood is actually considered a connective tissue, which is pretty crazy. Anyway, so that kind of covers it for the main, like the very basics for the cells, organelles, and the basic of tissues. Alright, so now I'm gonna go into the prokaryotic cells. Uh, excuse me guys for 1 second. I got a phone call. How do I pause this?